Hello students, welcome to Raaonline. In our question based brief discussion of topics at the level of need, our chapter is cell, structure and function. Cell structure and function, NCERT 11th syllabus. And let's start. Cell, that is the study of cell is cytology. Study of cell is cytology. And cell was discovered by Robert Hooke. Cell was discovered first by Robert Hooke. He was English botanist, discovered cell in 1665. He discovered cell in 1665 and he was English botanist. He discovered cell from cork, a plant product. He discovered some cell from cork. Observed under microscope, he saw honeycomb-like chambers. And for those chambers there is a Latin word cella. So word cell is derived from the Latin word cella, and that word is also given by Robert Hooke. So as far as discovery and name concerned, Robert Hooke discovered cell and gave the term cell also. Robert Hooke discovered cell and gave the term cell also. But there is a drawback. Robert Hooke discovered non-living cell. Actually, cork is non-living. So he discovered a non-living cell and it was in group. That is, it wasn't independent cell. So that's why independent cell was discovered first by independent and living cell was discovered first by Anton von Leeuwenhoek. So in this question, the statement 1 is correct. Then this is the definition of cell, that is a structural and functional unit of life. It's a structural and functional unit of life. Then life is possible when the body is made up of cell only, at least a cell. If body is cellular, then organism is with life. So this statement is also correct. And Schleiden and Schwann, German botanist, Schwann, German geologist, they formulated cell theory. They formulated the cell theory. So Schleiden and Schwann, they are known for the cell theory. So finally, in this question, that is question number 1, that is, we have to select the options. 1, 2 are correct. No, all these are correct. Best answer is answer number 4. I would like to add one more thing, that is R. Virchow. He modified cell theory actually. And he actually added one more statement in that cell theory, that is regarding origin. R. Virchow stated that hominis cellula e cellula. That is, every cell originates from pre-existing cell. Hominis cellula e cellula means every cell originates from pre-existing cell. And this statement was added by the Mr. R. Virchow. Then who concluded that based on his study on plant tissue? Actually, Schleiden was botanist. Schleiden was German botanist and he studied the plant cells only. and who concluded that plant tissue, that is presence of cell wall, is the unique feature. And Swan was the German zoologist. He was German zoologist. He was German zoologist. Though the German botanist was Sweden, but Swan studied number of animal cells and compared the animal cell with the plant cell and concluded that is cell wall is the unique character. So this statement was actually of Theodore Swan. And R. Virchow I have already mentioned. He stated that omnis cellula acellula, that is every cell originates from pre-existing cell. Whereas Robert Hooke, he discovered cell from cork. But he discovered non-living cell and it wasn't free. He discovered non-living cell and that wasn't free. Now, question number 3. Question number 3, that is prokaryotes are generally smaller than eukaryotes, right? Prokaryotic cell multiply slower. This is wrong statement. They multiply faster than eukaryotic cells. So, in this question, incorrect statement is, we have to choose the incorrect statement. So, this one is the incorrect statement, that is prokaryotic cell multiply. So, they multiply faster than eukaryotic cell. They are very simple type of cells, and they multiply very fast. They have only one option of their multiplication, that is amitosis. Prokaryotic cell multiply by amitosis only, and simpler mode of cell division. The mode of multiplication of prokaryotic cell is by means of amitosis. Then prokaryotes may vary greatly in shape and size, right? And the 4 basic shapes of bacteria, this is also right. These 4 basic shapes are, spherical coccus. Spherical bacteria is coccus. Rod-like bacillus. Rod-like shape of bacteria is bacillus. If it is rod-like, then bacillus. Then curved rod, that is vibrio. If it is curved rod, then it is vibrio. And the last one is coiled, that is a spiral. These are the 4 possible shapes of bacteria, out of which bacillus is quite common shape. Most common shape in bacteria, that is rod-like, and rod-like is known as bacillus. Question 3 is now over. Question 4. Question 4. This is the based on the comparison of plant and animal cell. This question is based on the comparison of plant cell with the animal cell. An organelle present in plant cell but absent in animal cell. Large central vacuole is found in plant cell. Ribosome is present in all types of cell. Ribosome is the only structure which is found in all types of cell, that is prokaryotic cell, eukaryotic cell, plant cell, animal cell, fungal cells, found in all types of cell. Plastids again in plant cell only. Plastids are found in plant cells only. Large central vacuole is found in plant cell only. So there are 2 features, that is A and C. A and C, these 2 are the structures found in plant cell but not found in animal cell. Large central vacuole and plastid, pigment bearing cell organelle, and it is one of the type is chloroplast. Suppose we imagine this is a plant cell. This is plant cell. Then you see large central vacuole in mature plant cell. Large central vacuole large central vacuole in plant cell. This is the large central vacuole. It occupies more than 90%, approximate 90% space. Approx 90% space of plant cell is occupied by the large central vacuole. And this is the covering of central vacuole known as tonoplast. The covering of central vacuole is known as tonoplast, and within the vacuole tonoplasm is present. Within vacuole tonoplasm is present. It is also called cell sap. Tonoplasm is also called cell sap. Tonoplasm is also called cell sap. So answer of the question number 4 is 4. Then next question is concerned with the prokaryotic cell. Many bacteria have small circular DNA molecule and they have outside the main genome. So this is suppose bacterial cell. This is a bacterial cell. It is prokaryotic cell. It has large double-stranded circular DNA as main genome. Then, in addition to this one, number of small DNA molecules are also found. Number of small DNA molecules are also found. This is nuclear equivalent. These are extra nuclear DNA. These are the extra nuclear or extra chromosomal DNA. These are extra nuclear or extra chromosomal DNA molecules. Extra chromosomal DNA molecule. So correct answer is plasmid. This mistake is often done by the student if they are in hurry. Though I know the correct answer, but in a hurry, by mistake I give the answer plasmid. That's why please be careful while attempting the paper. So correct answer is plasmid. Correct answer is plasmid, not plastids. Desmids, they are kept along with the diatoms. Desmids, they are kept along with the diatoms. Now sixth question, mesosome. In bacterial cell, this is cell wall. This is the cell wall of bacterial cell. The next to cell wall, cell membrane is present. Next to cell wall, cell membrane is present. This is cell membrane, and you see some folded structures are formed by the cell membrane. These are called mesosome. These are known as mesosome. These folds of membrane towards inner side, they are known as mesosome. These folds of membrane are called mesosome, and they are common in gram-positive bacteria. Common in gram-positive, rare in the negative. And they perform various activities like, see this one. Number 1, cell wall formation, right answer. DNA replication and distribution, it is also right answer. They help in respiration because respiratory enzymes and electron transport system is located in this membrane, and this folds give more area to the cell for that process. Hence, all three are correct. That's why my answer is question that is fourth. This is mesosome, folds of membrane towards inner side in bacterial cell. Seventh question, polysome. Polysome is also known as ergosome. They are called ergosome. Polysome is also known as ergosome. They are also called polyribosome. They are also called polyribosome. At the time of protein synthesis, at the time of protein synthesis, 4 to 6 ribosomes are attached on a thread of messenger RNA. 4 to 6 ribosomes are attached on a thread of mRNA. This is messenger RNA thread, and these are the ribosomes, and their sedimentation rate is 70S. So, number of ribosomes are attached on a thread of messenger RNA, but only at the time of protein synthesis. That structure is known as polysome or polyribosome. So, several ribosomes, they are attached on a single thread of mRNA, and at what time? At the time of protein synthesis. At the time of protein synthesis. This is seen at the time of protein synthesis. Eighth question. Gas vacuoles are found in the blue-green algae, purple and green photosynthetic bacteria. Their role. Actually, when gas vacuole is formed, suppose this is BGA, and in this blue-green algae, this is the gas vacuole. If it is formed, then this cell come to the surface of water. That is, it come to the surface of water. When they come to the surface of water, this vacuole ruptures, gas is released, then they are back again inside the water. That is, back again inside the water. So, these vacuoles are basically control their buoyancy. That is, when it is formed, cell come at the surface. When they rupture, gas is released, they are back inside the water. So, this is called buoyancy. This is the role of gas vacuole in cyanobacteria, that is blue-green algae, as well as photosynthetic bacteria also. Question 9. In membrane, lipids are arranged within the molecular. Regarding the structure of membrane, fluid mosaic model was given. The most authentic model regarding the structure of cell membrane is fluid mosaic model. This model was given by Singer and Nicholson. This model was given by Singer and Nicholson. And according to this model, lipid is bilayered. That is, membrane is mainly composed of lipid, that is lipo, plus protein, that is proteins, in which 40% part is of lipid and 52% part of the protein. This analysis was done in RBC membrane. Rest less than 10% is carbohydrate, less than 10% carbohydrate. And membranes are selectively permeable in nature. This is the nature of membrane. Selectively permeable. Membranes are selectively permeable in nature. And average thickness of membrane is 7.5 nm. These are some common features. And membranes are found in all cells, that is, they are in prokaryotic cell as well as in eukaryotic cell. One more thing, that is, in eukaryotic cell, membrane bond cell organelles are also found. So, membrane in eukaryotic cell, that is just below the cell wall, first one, and second, it is also around the cell organelle. Most of the cell organelles are membrane bond. As far as its structure concerned, the membrane is made up of two layers of lipid. Membrane is made up of two layers of lipid. that is bilayered. Lipid is bilayered in membrane. And if we see the molecule of lipid, it has hydrophilic head and two tails. They are hydrophobic. And their orientation is, that is, head is towards outer side. Head is towards outer side, whereas tail is towards inner side. So polar head, polar head, and non-polar tail. That is membrane. Lipids are arranged, that is polar head towards outer side and non-polar tail towards inner side, and tails are hydrophobic. Tails are hydrophobic, so this is also correct and this is also correct. That's why my answer is B and C. B and C. Tail facing orientation of two lipid layer. Tail facing orientation of two lipid layer is seen in the membrane. Tail facing orientation of two lipid layers. Fluid nature of membrane. Fluid nature of membrane. It is helpful in growth, it is helpful in secretion and helpful in cell division. That's why answer is 4. That is, fluid nature of the membrane is important with respect to all these. Now question number 11. Through membrane, movement of molecule with and without the requirement of energy. With and without the requirement of energy. With requirement active, without requirement passive. With regards to the active, if energy is required, then active movement. Energy is required in case of active transport, whereas in passive transport, without the requirement of energy. So first one is correct. Then with requirement, uphill transport, and without requirement, downhill. So statement number 2 is wrong. First and third is correct. That's why answer is 4th again. That's question number 11. 4th answer is correct. Question number 12. In fluid mosaic model, one of the following statement is correct with respect to the movement of lipids and protein. Both lipids and proteins can flip-flop. Wrong. While lipids can rarely flip-flop, protein can. This is the right answer. Lipids rarely flip-flop. What does flip-flop movement? That is, these are the two lipid bilayer. These are the two lipid bilayers and two molecules of lipid layers. This is in one, that lipid is bilayer, two layered. So this is of one layer, this is of the second layer. And this moves toward this side, and this lipid molecule come to this side. This is flip-flop movement, but it is rarely seen. It is rarely seen. That's what the answer is. Answer is 2. Cell wall, cellulosic. Cellulosic cell wall is feature of plant cell. But here galactans, mannans and minerals and calcium carbonates are there. And these type of elements are seen, these type of compounds are seen in the algal cell wall. Plant cell wall, cellulose, hemicellulose and pectin. In plant cell wall is made up of cellulose, then hemicellulose, and very small amount of pectin. This is about the plant cell wall. Fungal cell wall is made up of chitin, no cellulose. That is non-cellulosic and made up of chitin. Bacterial cell wall is also non-cellulosic, made up of mucopeptide complex. Mucopeptide complex. So that's why algal cell wall is correct answer. Algal cell wall is correct answer. Cell wall is non-living layer and it is permeable also. Middle lamella. In plant cell, generally cell wall has two layers. Cell wall has two layers. This is primary cell wall that is present at initial stage. Then in mature cell wall, this primary wall is replaced. This primary wall is replaced by the secondary wall. This is secondary wall. It takes place of primary wall but only at maturity. But when see two adjacent plant cell, this is plant cell number 1, this is plant cell number 2. These two cells are compactly arranged. These two cells are firmly joined together by means of a hydrophilic substance, that is calcium pactate. This is middle lamella, which is responsible for the joining of two successive cells. That is a common substance present in between two cells which fixes firmly. This is middle lamella. It is also termed as intercellular substance. Middle lamella, that is, it is also called intercellular substance. Middle lamella is known as intercellular substance. Firmly fixes to adjoining cell. Firmly fixes to adjoining cells. So it's an adhesive material. It is adhesive material. If it is dissolved, cells get separated. But its chemical composition, that is calcium pactate. Calcium pactate is the main ingredient of middle lamella. Though it also contains magnesium pactate also. Magnesium pactate is in a smaller amount. Magnesium is also present but in a smaller amount. Mainly calcium pactate, responsible for the fixation of two adjoining cells. Plasmodesmata. Plasmodesmata, actually it is present in between two adjoining cells. It has a protoplasmic strands. Protoplasmic strand actually. It's a protoplasmic strand present in between the two cells. It is responsible for the connection between two adjacent cells. Protoplasmic strand. This plasmodesmata is actually protoplasmic strands. So communication takes place in between these two cells. These two cells communicate through plasmodesmata. Main arena of various cellular activity. Answer is cytoplasm. Cytoplasm contains all the cell structure. Cytoplasm contains all the structure and it is the main arena of cellular activity. Nucleus is controlling center of cell only. Nucleus is controlling center of cell according to J. Hammerling. Nucleus is controlling center of cell according to whom? J. Hammerling proved that. J. Hammerling experimented over acetabularia. He experimented over acetabularia algae and concluded that nucleus is controlling center of cell. Plasma membrane is found in the all cell. It's a living structure. Mitochondria It is found in the eukaryotic cell, plants as well as animal, and it is related with the respiration. It is related with the respiration. Then which of the following structure are not considered as part of endomembrane system? First of all, endomembrane system. Number of cell organelles are found in, number of membrane bound cell organelles are found in eukaryotic cell. First, that is membrane bond cell organelles are found in eukaryotic cell. Membrane bond cell organelles are found in eukaryotic cell. Not found in prokaryotic cell. Then some cell organelles, their function is coordinated. There are some cell organelles like endoplasmic reticulum, Golgi body, lysosome, vacuole. They function in coordinated manner. And those cell organelles whose functions are coordinated, they are part of endomembrane system. Those cell organelles whose functions are coordinated with each other, they are part of endomembrane system. But there are three cell organelles. One is chloroplast, second peroxisome, third mitochondria. Their function is not coordinated with those. So these are not the part of. Question is negative question actually. Not considered as the part of endomembrane system. Answer fourth is right. Then endoplasmic reticulum. Endoplasmic reticulum in a cell. This is plant cell or animal cell. Any one cell is there. This is nucleus. And you see a structure in any cell that is highly present in the form of network. This is endoplasmic reticulum. And endoplasmic reticulum divides the cellular space into two parts. The space within the ER is luminal space, and the space outside the ER is extraluminal space. So this is extraluminal space, space present outside the ER is extraluminal and within ER is intraluminal. So that's why it divides the intercellular space into two parts. Divides intercellular space into two parts, that is luminal space and extraluminal space. So the answer is first one. Animal cell, lipid-like steroidal hormones. There are two types of ER. One ER on which ribosomes are present on their surface. They are called rough or granular ER. And because of presence of these ribosomes, these type of ER are mainly found in the cells which are involved in protein synthesis. Whereas those endoplasmic reticulum devoid of ribosome, that is ribosomes are not present on them, they are agranular or smooth ER. So there are two types of ER, that is granular, agranular. Agranular ER are mainly concerned with the synthesis of steroidal hormones. So answer is agranular ER. Agranular ER, they are concerned with the synthesis of lipid-like steroidal hormones, especially in the animal cells. We can also call them smooth ER. They are also called smooth ER. Smooth endoplasmic reticulum. 20th question. ACR, RER, ACR that is a smooth, are agranular due to absence of ribosome. Then RER that is rough or granular ribosome. They can be distinguished with its presence in. ACR and RER can be distinguished with its present in. And the number 4 is correct, that is ACR is present in those cells which are involved in the fat or lipid like substances synthesis, whereas RER is present in protein synthesizing cells. So the answer of this question is 4, that is fat lipid like synthesis is role of ACR, whereas RER are involved in protein synthesizing cell, and this is the only option in which protein synthesis is given after the fat synthesis. Question number 21. How many of the following cell organelles are membranous? That is membranous cell organelle. Membranous cell organelle is feature of eukaryotic cell. Eukaryotic cell is known for double envelope system. Eukaryotic cell is known for double envelope system. That is cell as well as cell organelles both are membrane bound. Whereas prokaryotic cell is known for single envelope system. That is, there is only one membrane bound cell organelles are not found. So single envelope system is found in prokaryotic cell. But in eukaryotic cell, there are few structures which are not membranous. ER membranous, nucleus membranous, lysosome membranous, vacuole membranous, ribosome non-membranous. And centrioles in animal cell, they are also non-membranous. So the answer is, that is, how many cell organelles are membranous? Then count this: 1, 2, 3 and 4. So final answer, membrane bound cell organelles 4 and non-membranous 2. The membrane bound cell organelles, their number is 4. 22 question. Which one of the following is responsible for mechanical support? Protein synthesis, enzyme transport. Best answer is endoplasmic reticulum. Endoplasmic reticulum, they are responsible for the formation of endoskeleton within the cell. They are responsible for the formation of endoskeleton within cell and provide mechanical support also. Then they have ribosomes on their surface, so involved in protein synthesis. And through ER, material is transported to the Golgi body. So answer number 4 is for the question number 22. Glycosidation, glycosylation. Glycosidation, that is linking of carbohydrates with lipid, is glycosidation, and linking of carbohydrates with the protein, glycosylation. And enzymes for these activities are found in Golgi apparatus. That is, Golgi has enzymes. Golgi body has enzymes. Those enzymes are concerned with the linking of carbohydrate with lipid as well as linking of carbohydrate with the protein. Such activities are called glycosidation and glycosylation respectively. cell organelle performs a function of packaging. That is, ribosome synthesizes enzymes over ER. These enzymes or proteins are given to the rough ER. From rough ER, they are carried out by smooth ER to the cis part of Golgi body. Cis part of Golgi body. And the Golgi body responsible for the post-transcriptional changes of those protein or targeting material, and they are released from the trans side. They are released from the trans side. So the packaging work and modification work is done by the Golgi body. The packaging work and modification work is done by the Golgi body. The question number 25. Number of proteins synthesized by the ribosome on ER are modified in. Just in the previous question, I have already given the answer. The answer is cisternae of Golgi apparatus. Cisternae of the Golgi apparatus. They modify the enzymes or proteins synthesized by the ribosome, and they are released from their trans surface. That's all. Thank you.